Welcome to Shastra Arambh, the show where innovation meets purpose. In Shastra Arambh, we talk to founders, tech enthusiasts, innovators, and leaders who share their inspiring journeys, opinions, and vision. Today, we have with us Dr. Rajnikant Vangala, the co founder and CEO of Neuome Technologies. Neuome is a biotech startup with proprietary technologies that have wide applications in the molecular diagnostic space. It's one of the startups that have also been featured in the Shaastra Magazine recently. How have your background and previous experiences helped you in building Neuome Technologies? Could you tell us a little bit more about it?
Thank you so much for having me, Pratibha. It's been a very big, very big thank you to you because, this is almost my first time to give an interview to anybody other than my company people. I do not know how to do that. So if there is anything that I don't know, please correct me. And so, yes, I have always believed in a very interdisciplinary and cross-disciplinary approach of doing science, and that's very clear from my own experience that I have done. And I'm actually a graduate in biophysics from Nizam College in Hyderabad, and
Okay.
chemistry. So biology or science, biological kind of side was fairly less, more
I see.
about physics and chemistry. And coming from that background, I knowingly chose to get into biotechnology because wanted to get into something more applied rather than pure, pure science. And thanks to University of Hyderabad, I did my masters there, and then I worked in NCBS. In those days, it was in ISC campus, way back in 97, 96. So I did my JRF with scientists in NCBS, and there I was working on Drosophila genetics. So coming from a very very different background, I have seen how the science transcends across different aspects, how the technologies transcend over technology. So you take an example of PCR machine. It is a physicist who made it. It's not a biologist. Biologists use it, right? So in that's what gave me an idea that we should always do an interdisciplinary approach of learning, which would then enable us to create newer solutions. So that's why when I went to my PhD, it was more on a clinical application. So wanted to understand how leukemia develops and bone marrow transplantations. This was way back in '99. I'm talking about when I PhD, and we were doing stem cell work and bone marrow transplantations and how some of the molecular factors influence the differentiation and how the disease comes in. And following which, I worked in Karolinska Hospital in Stockholm, and also in Imperial College London Hammersmith Hospital. And so going through all these scientific endeavors in international labs completely tells you that the focus of a specific field should only come when you have baseline understanding of different aspects of different fields of science. So that is how this company's formation, we call the tagline as convergence of science.
Right.
You see, so different fields of science come together to create a solution, and in that is what our expertise is. We are R&D scientists. Our company is only R&D. We have different divisions, molecular biology, genomics, proteomics, nanotechnology, microbiology, and biomedical engineering. We have an IT department. So it's actually any product which gets developed goes through all these aspects. So that is what actually creates the innovation.
Right.
And innovation comes in when you create, and so anybody who wants to create a parallel product, the entry barrier becomes very high for them
Right.
because we've used a very cross-disciplinary approach of building the technology.
Right. So I understand that you set up the company right in the middle of COVID in April 2020. So what was that period like for you, and how did your team? What was your team's approach towards that situation?
So we registered the company on April 16th, 2020, and 2 or 3 weeks later there was a complete lockdown.
Correct.
And the company was formed with a basic idea of fostering, developing, and commercializing a product called InstaPreserve. But the moment COVID happened, we did not have any wherewithals of knowing what the hell is going to happen. So
Right.
eventually, we basically we went into a 1 month not knowing what to do during the lock in time, lockdown time, and what we came up with during that time, with lot of brainstorming discussions and all, is that the need of the hour is point of care diagnostics. We may have a great technology called InstaPreserve for sample collection and all, but the need of the hour seems to be point of care devices. So
Mm-hmm.
we immediately started doing research on that. We developed a platform which uses peptides as the base of diagnostics, peptides and nanotechnology. And gold nanoparticles were designed, peptides were designed, synthesized. They were used in diagnosing COVID.
Right.
So we immediately formed a separate entity in Singapore, which took over this product called InstaDetect as a platform.
Mm-hmm.
And we got investors at that time for Singapore entity. We raised $5 million immediately, and we got the product developed, and it was approved by ICMR. So we developed the products using this peptide technology for COVID detection from saliva, from breath, and even from the nasal swabs. So we got all these three approved by ICMR and Cisco. So that was one part. So the InstaDetect platform came in during the time of COVID as a need for addressing the need at that time as a point of care. But then what happened is point COVID went off, but point of care became a very big need in the healthcare now, and I think there is a very wide recognition in at the government levels as well as in the private hospitals that the need of point of care is very high. We need to implement this, and so we are using this platform in developing several metabolic disorders, cancers, infectious diseases assays as a point of care diagnostics. For an example, we are now coming up with troponin I as a system with a finger prick, just like a diabetes test. So what this enables is, if somebody is feeling very uneasy at home, you may not know whether it is because of the gastric problem or it's because of actual cardiac event. So you can immediately take this and test it out. And if don't have at home, you can actually call an ambulance. While coming in ambulance, they can test it in ambulance. you know and after coming in, if you are in an emergency ward, they're also there. And the beauty of this is that it's not a qualitative assay, it's a very quantitative assay, and the assay time is within 8 to 10 minutes. So all these advantages, that and you don't need any special training to use this.
Right.
So all these advantages make our InstaDetect a very, very useful point of care diagnostics technology, and that is coming through our Singapore entity joint venture that we have formed. While we were forming this and everything was happening, the Indian company continued working on InstaDetect.
Right. So, what about InstaPreserve? That was another product that you developed that was already developed back in April 2020. You said, how has it come about, and what other products do you have in the pipeline right now?
So the origin of InstaPreserve technology per se was my experience in Thrombosis Research Institute. I was director of that institute for a very long time, and it has two branches. TRI is in London and in India. The Indian branch was essentially closed once Professor Vijay Kakkar, the founder chairman, passed away um, in 2016. So 2019 or so, they were actually planning to close down. And the problem with such a situation is that we had a very large biobank of 15,000 patient samples. And all these patient samples had to be discarded. And that's a tremendous loss for any biomedical scientist. No clinical research should ever discard any sample.
Correct.
So I do not know exactly whether all of them are discarded or not, but that kind of a difficulty arose primarily because sustaining a biobank is a very costly affair. So just the electricity bill, just imagine electricity bill for 20 different -80 freezers. I don't think even a private company will ever do that, you know, unless they are making some money out of it. And you cannot
Right.
make money out of biological samples. So, what was more important to me was that in a country like India, we do not have large genomic studies. Why? Because we can't afford to have large biobanks. It all boils down to money. We don't have money. It's as simple as that. So if you want to break that, you need to develop a very cost-effective technology, and which can remove the cold chain. So with this intention, in 2017 itself, I started research in parallel, and then I formed an institute called Institute for Applied Research and Innovation, where the actual research started and continued, uh, where we have identified that a specific cocktail of three or four chemicals can sustain any biological sample for a very long time. And we have done some enhanced stability studies after putting the samples and all to show that for 15 to 18 years, the samples stay very intact. You can isolate DNA, RNA, protein, or do all the other diagnostics. So it really helped us that we developed a solution uh, for collecting the samples and storing for long term. But after we formed the company, for commercialization, you need a separate set of assays to be done and validated. So we have applied for a DBT grant, big grant, and we got selected, and we are a big grant awardee. And using that grant, while the InstaDetect was getting developed, we had a separate division and separate bunch of scientists who were working on InstaDetect, sorry, Preserve, and they then built it into a platform technology. So what has
Mhm.
happened is now, with this grant and help from some of the investors, we have made it into several aspects. What initially happened was that this sample was sent to several friends who had different diagnostic labs in across the country, and one of our friend who is a pathologist, histopathologist, he said, I would like to see whether it can fix a given tissue or not. So he took a thrown away sample and then put it in there, and he did histopathology. And he showed that actually this solution works far better than formaldehyde,
I see.
which is a very toxic chemical, very cheap, of course, but very toxic chemical. And
Mhm.
it is the need of the hour if you want to change the toxic practice that we do in all the pathology labs. So then what happened? How do you make it cost effective? So now we have a product called InstaPreserve. It can be used for DNA isolation. So that's a separate SKU that we made, which is called Nucleosafe. And we devised a newer formula for RNA. We call it RNA safe. Like you have RNA later and all, where the limit of time is that 14 to 15 days, but with ours it is years. RNA is stable for years, and immunofluorescence. And then we have developed another SKU for proteomics, InstaPreserve Protein Safe. Then we now have different SKUs more called tissue safe, which is for histopathology,
Okay.
and med safe for anatomy.
Hmm.
We can actually store the entire organ uh, into the into the solution in anatomy labs.
Interesting.
In medical colleges and universities where they preserve all the organs in formaldehyde, 10% formaldehyde. Now you don't need to use that formaldehyde. You can use our solution instead, which is
Right.
100% green chemical, ecological friendly, and non-toxic. And biggest advantage is that it is the same cost as that of formaldehyde. So now we built a very cost-effective solution which has a very wide application from molecular diagnostics, biobanking, to histopathology, to anatomy, and now recently, with help of some hospitals in AIMS hospitals, we have shown that actually even in cadaver embalming, we can
Oh,
replace formaldehyde with our solution Instapreserve. So it really has come out. As a scientist, I only wanted to work on my biobanking as a main thing, but users, new and new users started to come. Like histopathologists said, we will use it for histopathology. Anatomists and forensic medicine people said we will use it for embalming. We will use it for something. So actually, users have developed more and more uh, products out of this platform. So in fact, even though I might claim that we started the research, but in fact it is the users and the clinical community which has come forth in making more and more products out of this.
right.
And every day we are seeing a new thing. So just uh, we are finishing one product which is called um, IP Bio, which is
Okay.
basically is for, so if you collect any blood sample you will have EDTA citrate and normal blood. So EDTA and citrate are used for serum and plasma, so that you can go for automated analysis. But when you collect a blood sample you need to put it in cold chain.
Hmm.
You have to use cool packs or you have to use something cold so that you can carry the blood samples. So we developed a cocktail now in which once you collect the blood samples you don't have to do any cold chain, and the samples remain completely intact for up to 1 week.
I see.
So even if you want to revisit the same sample you can actually go back and revisit it. And
Okay.
that's a very significant change in the entire in the molecular diagnostics and biochemistry, pathology, histopathology, and everything. So what we thought that InstaPreserve is for biobanking, it has spun out to be a product, a technology which has come out with multiple utilities for multiple clinical faculties.
Yeah, in fact, that is one of the questions that I wanted to ask you as to where exactly, which are the sectors exactly that you have, your products have found applications in, which you've explained so far. But I also understand that it has some amount of applications in veterinary sciences also. Could
Yeah,
you
yeah.
explain that a little bit? Yeah.
Sure, sure, sure. I mean, in fact, being clinical scientists that we are, we know that we can't directly jump into human samples.
Right.
Right? So we could the easiest is to go to a butcher shop and get an animal sample, right? There is no ethics
Mhm.
approval for that. So
Right.
because you need to follow rules and norms of the land,
Mm.
you should not do anything which is ethically wrong. So we started off with a lot of animal studies,
Mhm.
and even that we actually got approvals, mandatory approvals and everything. And we also published now that in different animal models, InstaPreserve works in all these aspects that I just mentioned.
Right.
And it's not just us; it's actually the users who are actually publishing this. Like Basavatarakam Indo-US Hospital in Hyderabad, they are publishing a 45 or 50 sample study that they have done. It's not we who are doing it, and they found it far better than formaldehyde. And
Okay.
that's the beauty of it, that it's not we who actually developed it; it's pathologists who developed for pathologists. and pathologists are coming forward to publish it by themselves, and that's the beauty of it. In animals too it is the same. Like the lot of veterinary doctors, I am an animal lover. We used to have a lot of animals in our farm, and we have a lot of friends in veterinary field. So they came forth, they started using this. We have collected several animal samples in the past 4 years. We collected
Mhm.
so many. We have a museum of animal samples and insect samples, and several things. So it's come out very nice. And another advantage is that it's also useful in plants and agriculture. So
I see.
usually, if you take even as an example, a plant sample, if you want to store, usually dry leaves or seeds, they are stored. But you can isolate a reasonably okay quality DNA, maybe, and that
Mhm.
too is very variable, and not everywhere you will get a good DNA. And that's why even plant biobanks need cold chain.
Right.
So we have removed that. We have started seed biobank, plant seed biobank, and we will be seeking out for collaboration with several medicinal plants institutes and horticulture and agriculture institutes to form India's and world's largest plant biobank without the need of cold chain. So whether
Right.
it is human, whether it is animal or plant, or even environmental samples. So we recently traveled to an area near to Uti, and we collected the soil samples where Adivasis live, the local tribes, and we took the soil samples from the near to waterfalls and to the agriculture side. And then when we came back, the samples which were stored in InstaPreserve had the microbial flora well sustained and maintained to know the microbial cultures of it. Everything was retained, and the samples which were not in InstaPreserve actually lost more than 70% of microbes.
I see.
So I think even an environmental sample, or even a plant sample or an animal sample, they are absolutely fine. So we have developed a huge library of that. And so if you look at human health, or even animal health, or any health system, we are an interaction with our surrounding. We cannot improve our health without improving our surroundings. So any large scale studies to make human health better has to have an integrative approach of environment, animal, plant, and human. So we are building now a largest biobank possible without the need of cold chain for all these aspects. So we will be traveling all across India and the world, if possible, to build world's largest biobank for human, plant, animal, and environmental samples.
Very nice to hear that. I'm also going back to what you said about the interdisciplinary approach that you take at Neuome Technologies. You mentioned that. So, what exactly was the gap that you found in the life sciences space that you were seeking to address back when you started Neuome Technologies, and how has the interdisciplinary approach really helped in addressing that?
I think majority of the diagnostic practices have not changed. They have remained the same for past 70, 80 years. Even during the COVID time, we were only talking about PCR, PCR, and PCR, but would never come out with something novel. We are scared. We are worried about something new. And if something new comes, you need ABCD valuation, validation, so on and so forth. And this becomes the biggest entry barrier. So you want to build something new, there is a huge entry barrier, regulatory aspects and all. And that's good. One needs to have good regulatory environment and controlled way. And that's why you get good quality products in the Western countries. But now India is caught up with it and has a fantastic regulatory regime, and things are coming up very good. What triggered me is the need of disruption that we have built in. An example I tell you, I mean, no matter how many people I talk to change people from usage of formaldehyde to non-formaldehyde, non-toxic chemical for fixation, not many people are ready.
Mm.
That's because for past 150 years they know formaldehyde works, even though it is toxic, even though it is not good for health, even though it is, but we know that it works. It's like it's been there for 150 years. So we don't want to change that. We don't want to change something which is working.
Mm.
But you cannot become sustainable if you are not disruptive. And the disruption must come in at some point of time. If there is no disruption, then it is we are happy with complacent. Then the society is complacent. A complacent society will never improve. And so what I saw, the need was the need for disruption.
Mm.
And the disruption should actually eliminate two major things. Number one is the price. We are a very price sensitive country, and that is, I mean, that should not be an excuse for quality.
Right.
Okay, so we should be able to provide the same, or actually the better quality than what we import. Healthcare, majority of it is import,
Mm.
and we import because we cannot make quality products that you get in West, right? So the main reason for that is no R&D is done for Indians by Indians. We do R&D when we do a PhD, and then you come back to India, we still continue the same research because we are scared that if I change my field now, will I get this publications, will I get my tenure, everything. So we don't want to change that. We want to stick to what we are done in PhD. And most likely we have done PhD because the Westerners need to work on their diseases. But when you come to India, this is what happened to me, that when I came to India, in Thrombosis Research Institute, it works on cardiac diseases. But of course,
Right.
thrombosis is a phenomena which is associated with cancers also. In every cancer there is, and in cardiac disease also. But we were initially focusing on cardiac diseases. So a lot of people told me, Rajni, you are changing your field. People will not accept you. You need to stick to your field, and this fear is what is the most important thing. That if you want to disrupt a field, if you want to bring something new, you need to get rid of your fear and start creating technologies for India, by India, in India. If you do that, you are creating a solution for 1.3 billion people. We are one third of the world's population. Rest two thirds will follow anyway.
Right.
If you create for one third of world's population, rest of the world will follow you. I believe in that. And so we started to work on high quality, better quality than what you can get in any Western country. I have lived in Western countries and I know the quality there. We are here to make a better quality than what rest of the world can ever create. We will create disruptive technologies which will really show to world that India can and India will make it, and we will then deliver it into the market. In this journey, there will be hundreds of difficulties. That's taken for
Right.
granted.
Right. I also wanted to check with you, since you've also worked in the West, and you have a comparative knowledge of how technologies work in the West versus in India. What do you see are the hurdles for startups like you, who are in the life sciences and biotechnology space, and how can it be addressed so that more such startups can flourish in India?
I think India is changing very drastically in that sense. In 2008, when I came back to India, I started a company called Innova Biologicals in Hyderabad, which was at that time located in ICICI Knowledge Park. But I could not raise money, you know, because being a scientist, there was no investor who was interested in molecular diagnostics at that time. I had to basically close down the company. And this is the problem because Indian investors typically would like to invest in mobile apps, in gold, in real estate, whether it is a middle class family or a HNIs to whoever it is. The risk appetite is extremely low in India,
Right.
extremely low. So fundraising became a very big difficulty in India. Even now, it is the same. Not much has changed, but of course, because of government's initiatives, like big grant that I'm so lucky to get, I say that the people are looking at and saying it is worthwhile to invest. But the biggest challenge of this part of technology is that it is highly regulated, and lot of regulatory norms are not completely understood in India. Regulatory compliances and this thing, and the investor are not really still there. I give an example of myself. I have been on fundraising for past 1 year or so for Insta Preserve. Uh, the many people whom I have met, and all that they would say is, "You start making money, then we will invest."
Okay.
Whereas in any Western country, including in Europe or US, you have an idea, and I think um, reasonably good idea. They will of course evaluate whether this idea is really novel, there is a need, and everything, and then they do not mind taking a smaller risk. But in India, no, no way, no way. And I can give a list of almost I spoke to around 65, 70 investors in India.
Okay.
I personally felt that these are all real estate people. They
Mm.
want to invest in. If you are making money, I will put money to multiply that money. But I
Okay.
don't care which technology you are making, how much you have suffered, and all that. That is not an interest for them. I'm not saying that everybody is like that. There are many investors who have, I mean, especially people who have done diagnostics, who have done this. They are very open. They come forth, and right now we received a 500,000 investment. All it is because of people who know the field. But then, how many people will know biotechnology? How many people will know healthcare? How many people will know diagnostics? This is the challenge. In India, I believe investor psychology has to change, and same should be the innovator psychology. Innovator has to come out with better innovations, not copycats, not to copy and then say that I want innovation, I have an innovation, I need money. Innovators have to come up with the better ideas and know how to pitch to people, and the biggest challenge is attracting good investment. That's the biggest challenge in India, and of course the regulatory.
Right. So, talking about your products right now, what exactly is your customer base like right now, and where do you see it expanding in the near future?
So one of our major customer base is in molecular diagnostics,
Right.
where any molecular diagnostic sample, when it is collected, a blood sample or a saliva or anything, you will have to freeze it down. There is no other way of shipping the samples. And that's where we see that we make a large impact, a very large impact, in fact. So recently we have we sold some product to Shankara Cancer Hospital. These were actually the people who initially validated that any biopsy sample they collect, they do remote area health screening. Earlier they used to take samples from remote areas in liquid nitrogen
Mhm.
and then bring it to the centralized lab and then do whole genome sequencing, so on and so forth. Now they don't do that. They just put it in InstaPreserve and they just simply courier it, and per sample your max cost will be like 7500 rupees. With liquid nitrogen there is nothing which is less than 1000 rupees per sample. So there is a huge cost save and huge value proposition in molecular diagnostics and large studies research. This is
Yeah,
one
especially in rural healthcare, if I'm
rural
not
healthcare,
wrong,
I
right?
think, and green technology, rural healthcare. they become completely accessible to a very large extent. Molecular diagnostics could never penetrate the rural arena, and now it can. So, lot of value creation there. Second one is the pathology, where this formaldehyde in majority of the labs is actually supposed to be. I mean, to tell you the honest truth, not many people, what you say, decontaminate it and dispose it.
Right.
Uh, you know, but the kind of toxic effects when they go into the lab, you can actually smell the fumes of formaldehyde. You know, and young students who are working, young women who are working in their early age get exposed to the most carcinogenic toxic chemical like formaldehyde. I don't think that we should allow our young generation, and especially the clinicians or the medical students, to be exposed to such a thing. I was writing an article called Pathologists Are Saving Lives, but who is saving pathologists' lives? Nobody.
Right.
Because these people are day in, day out exposed to formaldehyde. So that is a very big market that I'm looking at. It's a huge change that we need to bring in Indian mindset, that we are okay with formaldehyde. In Europe, for example, they have banned it. They have banned the usage of formaldehyde. You know, but because there are no alternatives, they still continue to a small extent usage of formaldehyde, and there are special designed laboratory areas where they should go and do it. You cannot do it anywhere and everywhere. Right? So I think that if the product is positioned well, maybe Indian market will accept to some extent, but the Western market is really looking for an alternative like this. So where they are banning the usage of formaldehyde in total. So these are the two major markets that we see that will be doing a very big uh, value.
Wonderful. I also wanted to ask you about your intellectual property rights and what are the initiatives that you're taking to protect your intellectual property rights, and how does that give you an edge in the market?
So most of these are patented. We typically, when we generate a complete proof of concept, we immediately file a PCT, and all of them are in the process of that. So we never leave any IP unpatented unless we realize and recognize that it is too simple a formula, it's too simple a thing to really patent. Then we do a trademark, we do a trade secret, and we make sure that the trade secret remains trade secret. And there are several mechanisms to do that. So I think uh, IP is a very important strategy, and we have roughly around 4 plus 6 plus 1, 11 patents in process. Four have been approved, four have been given, seven more in pipeline.
Right. I wanted to understand what are do you notice any trends in the life sciences and biotech sector, particularly, that are interesting and exciting for you currently, emerging trends that you can notice that particularly excite you? And how do you think are these trends going to impact your particular new technologies per se?
I think there is a very positive change. If you really look at the diagnostics per se, just a very small segment of the entire healthcare system, we are not even $100 billion. We are not even $10 billion.
Right.
Okay? But we are a billion strong people. So even if you say 1 rupee per person, we are a billion, or $1 a person, we are a $1 billion economy in diagnostics. So there is a huge potential to grow. There's an enormous potential to grow. It is need of the hour is innovators who can address the real challenges, innovators who can take up certain risks, and investors who are willing to risk a little bit too. And I think if and government is creating enormous and great opportunities to do this both. And there are several conferences, several aspects that the government has come up with, which is really helping. So I believe that Neuome Technologies will become a very pioneering organization in disruptive diagnostics. We will be creating many more platforms like this, and come out with newer products. And we really are very much positive minded in the growth of the market.
Right.
Yeah.
I also wanted to ask you about your training modules, genomic training modules that you have. How exactly are you looking at molding future scientists who undertake these modules, and what do you have to offer?
One of the most important things growing up uh, is that as a student, it is very important that you get a cross disciplinary learning. And once you take up something in your career, then you start focusing on it so that you become very good at it. So when students come to our company, because we have 5 or 6 different departments, we tend to expose them to different aspects of science, whether it is a nanotechnology, whether it is a molecular technology, whether it is NGS, or biomedical engineering, or so on, and then let them develop, come out with some ideas. We sit with them, we design something with them, we make them explore, learn. And failure is okay. We don't mind students not able to get some data,
Right.
because otherwise they will not know how to question why it didn't work.
Mhm.
So we actually welcome failure and say it's okay to fail, learn with failures. So our teaching modules primarily focus on that practical ability of a student to make them think more. They have to. And sometimes, in fact, we give reagents which don't have all the chemicals, or we make them do it and then we make them fail in it, and then ask them to question how to troubleshoot this. See, the science is not in results. Science is not in getting all working results. If you do not know how to do troubleshooting, you will never learn science. And you can only troubleshoot when you fail. So we teach them in a very problem based learning approach, and a very cross disciplinary approach. And they have the freedom to change from one to the other. And that only helps them to become stronger in whatever they select. As I always believe, a human being should know what they don't know, which is more important than what they know. So, a student should also be able to try everything and then say that I'm not going to be happy with nanotechnology. That's not my cup of tea. I'm going to be good in IT. I'm going to be good in, maybe I'm good in molecular biology, and that's what I want. But if you're not exposed to different fields, you will never know. So that's what we do. Our training modules try and focus students to improve, not just as a technique. you know Skilling does not mean you just teach them one technique and that's over. Skilling means changing the mindset of students and making them empowered with thought process of solving the problems.
Right. So there are many young people who are looking at taking up a career in biotechnology and genomics. Could you tell us a little bit about the intersections of technology and life sciences and the emerging trends in the sector that would encourage these students further?
Absolutely, absolutely. I mean, some of the most uh, latest aspects is the microbiome, for example. And microbiome seems to be one of the most important sector, because whether it is human health, whether it is a plant health, whether it is a animal health, it seems to be governed by the microbes within us or outside us. Majority of infections are the ones which are outside us, and also some of the stuff which are inside us, either we lack them or we have them in more. So microbes seems to make a very important aspect in our life. So coming with this latest thing, actually, students, if they can learn how to understand the microbiome, whether it is a oral microbiome, esophageal, or gut, stomach, intestinal, rectal, or whichever microbiomes, and build technologies and products around this. I think that we will be eating better food and stay healthy. So, I would say the latest, when you look in Instagram, Facebook, all these things, every fourth or fifth ad is about microbes.
Correct.
How your microbes are important for yourself, how you should eat good yogurt based products, and so on and so forth.
Probiotics, prebiotics. Yeah.
Probiotics. So I think that, and even, I mean, even though this kind of practice was always existing in cancer patients, right? When we do
Mhm.
radiation therapy or chemotherapy, we make sure that we feed them good yogurt. We make sure they get good microbes because we want them to be replenished. Because chemotherapy or radiotherapy, that kills everything. But then we recognize that actually we are eating enough toxins without having cancer enough. you know so we need microbes. So there is a lot of research on that, lot of work on that, and if students can come out, learn how to do this, I think employability becomes very easy. Because one, of course, you will know the genomics. You know how to analyze a sample. You know how to create a report. You know how to give a value that a doctor needs to talk to his patient, his or her patient. So I think learning genomics per se is not just about just doing one technique and going home. There is a lot more value add that you can do around it, as to how you can analyze it, give a value report for the doctor to take it forward with the patient, and that's what students need to learn.
What about your advice for entrepreneurs in the space of biotechnology, life sciences space, especially?
Never be scared of failure, and at every step you will keep failing,
Okay.
and every time you fail you just get up and start running, if possible flying. And to tell you very frankly, in the past 5 years of this company, we failed miserably. Miserably, we failed, and we came up from that. The only thing that matters is that you have right set of people along with you.
Right.
Investors is one part of it, but if your co-workers, if your co-founders, your co-workers, they are not with you, if they are only there for 9 to 5 job, or if they are not dedicated enough, then you just become kind of a drainage of money. So you need a set of people whom you can rely on, whom you can put your faith and say they will not let down the company, not you, the company. So when I started, it was a one man show. My co-founder was in Hyderabad all the time. I slowly built it to a 30 odd people company. And today, even if I told them that, if I hope that never happens, if I say that I will not be able to pay salaries for a couple of months, they will be absolutely fine. Because there was a time I was discussing this with them. How many of you would be leaving if I say that, a month or two or three, if I cannot raise the funding, if you want to go for a job, please go ahead. But I will hope that such a situation doesn't come. But if you want to find a job, you're most welcome. None of them left.
Right.
None of them left. And I think that's a faith reimposed, right? You have your team which puts faith in you and you put faith in them. It goes both the ways. So it's an extremely important thing to have a good team, a dedicated team, a team which puts their life for you and you put your life for them. That's what is the most important thing for an entrepreneur.
Right. My final question for you is what is your long term vision for your company?
I think we want to be the most disruptive oriented diagnostics company, bringing in a very, very um, innovative technologies which will break these huge barriers of needing large instruments, imported instruments, and specialized instruments. And typically, what I would say, I'm not against the Western or Eastern or any this thing, but science is all about simplifying things. Science is about simplifying our life, and it should enable us to live happy and better. But when science creates a huge big gadget like a minus 80 freezer, then you cannot afford it. Science has to make our life more affordable, more easy, more simple, so that we focus on quality of life. We lead a good quality life. And that will be our focus to break these huge barriers that we have put in the name of technology, large machineries to so on. Simple things are the best answers in life. So our idea is to bring simple, novel, disruptive technologies which will make India the front runner in healthcare.
Thank you very much, Dr. Rajnikant Vangala. Thank you for joining us and sharing your story with us today. We wish you all the very best. And for all those who are watching us today, I would urge you to pick up a copy of Shastra Magazine or visit us at shastramag.org and find out more about the promising startups that we feature in the magazine regularly. See you again with another episode of Shastra Aram. Don't forget to subscribe to our YouTube channel for more such interesting startup stories. Thank you.
Thank you so much. Thank you.
Thank you.
Thank you.
Thank you.